Hello, everyone. Welcome to our webinar, Accelerating Drug Discovery with AI Agents. My name is Sergey Ananyan, and I represent the company Megaputer Intelligence. Today, we'll be focusing on the use of generative artificial intelligence for target identification. This is the crucial first step in drug discovery, focusing on finding specific molecules that play a key role in a disease process and can be modulated by therapeutic agents. In the past, target identification was an art based purely on serendipity. We'll outline how to use AI to help researchers turn target identification into a well-organized process and significantly accelerate early drug discovery. We'll focus on the use of collaborating generative AI agents that enable the discovery of patterns of interest, eliminating hallucinations, and revealing chains of causation triggered by different pathogens. The process of target identification frequently involves processing enormous amounts of textual documents, as well as working with huge domain-specific ontologies. And this is why efficient solutions rely on the joint use of advanced natural language processing techniques used for quickly reducing the number of documents, followed by more in-depth analysis performed by AI models on the remaining potential target documents. We'll demonstrate a practical solution for target identification built on a no-code data processing and analysis platform, PolyAnalyst, manufactured by Megaputer. PolyAnalyst helps orchestrating the work of multiple AI models, as well as their joint work with NLP, data processing, and report generation agents. The user of PolyAnalyst visually creates the necessary data analysis scenarios using a large library of tools provided by the platform. Let me illustrate how this works. So here you see a fragment of a solution for early drug discovery that involves different large language models working in concert with each other. And you can see there are more models that are hiding on different next levels in this data analysis scenario. And in general, the user of PolyAnalyst builds multi-step data analysis scenarios from a large library of tools. Here you see a collection of tools that work as connectors to different data sources. It can work with databases, collections of documents, various images, JSON files, and many more. And frequently, before the analysis with AI agents starts, the data has to be massaged in the necessary form. So here you see various data processing tools. Then PolyAnalyst offers a large selection of text analysis tools based on NLP and also AI agents that we'll be covering in more detail. And these are used in combination with various machine learning engines, followed by visualizations. The results of the analysis can be exported to multiple formats, and the solutions can be automated with a collection of other tools that help building multiple loops, fix the sequence of execution, and do many other things like condition-based following of one or another branch of the analysis. Users of PolyAnalyst can quickly build solutions for multiple applications, and one of them we will be discussing today. So I have with me Sam Montgomery, a scientist who actually had build some solutions for using AI for drug discovery. So let's move on. Sam, my screen is seen, right?
Yes, I can see it.
Good. So what we are going to discuss are several topics. We'll focus on one of them. AI can be used for accelerating early drug discovery, first and foremost for biological target identification. So we will be looking for different targets like proteins, genes, pathways, and so on. It can be used for designing new molecules because the chemical space is enormous and people are finding new drugs right now by some serendipity instead of systematic approach. It is possible to predict absorption, distribution, metabolism, and excretion and toxicity because the new molecules that people are finding frequently have other difficulties that they have to overcome to make the drug effective. And also, it might be possible for repurposing existing drugs. So people are looking for new targets that they might affect and become efficient for treating new illnesses. So today we will be focusing on biological target identification, and this is frequently the first step in drug discovery process. We need to find what we can act upon to prevent the harm done by various pathogens. And this might be some cellular, some molecular structure involved in a disease that drug is supposed to work on. And we might want to focus just for the purpose of demonstrating the solution on the case where different pathogens are modulating the immune system response. So we would like to find the mechanism that they're using and then take advantage of this mechanism. See, for example, which illnesses we can treat with the help of knowing and using this mechanism. We can work on different results of research. One of these sources would be published articles that you can find cited on PubMed, for example. And the task looks simple at first, but if we're interested in finding information about all possible pathogens and, for example, different genes that can modulate the immune system response, the task quickly becomes overwhelming because there are tens of thousands of different known pathogens. There are millions of genes, and PubMed itself would contain close to 40 million citations already. So
So just to clarify, Sergey,
Mhm.
To make sure we've covered our bases, we're looking for cases where a pathogen is having an immunological or immunomodulatory response effect on the gene, with the idea that we can create an intervention that has the same immunomodulatory response using the same mechanism as the pathogen.
Yes, thank you, Sam. All right, so what would be a typical, so how it was done in the past? People would be searching PubMed articles to identify potentially relevant articles. They would focus on them. It involves lots of bias. Read them and then get ideas what they can use. So they would then generate a report for their colleagues, fellow chemists who can help them create the molecule that would affect certain targets, or when people have different molecules, they would identify what they can act upon. So what can be done to automate this process? So we will be demonstrating a solution that automates all three steps. So first, there will be linguistic queries that sift through millions of publications to detect patterns of interest. And when we're working with 40 million documents, tens of thousands of pathogens, and millions of genes, we really have to become very efficient in this process to get results obtained in some reasonable amount of time. The second step is using different AI engines. So it would be a multi-agent system orchestrated through a no-code system that helps automate the task of validating the tentative patterns that were discovered through linguistic rules, because linguistic rules are not perfect. And we also want to reveal target genes and exact mechanisms that pathogens are using for modulating the immune response. And the third step of automation, we want to publicize our results for multiple researchers, so they do not need to worry about all the complexities of the process. They just want to have a simple interface where they come, ask questions, and get immediate answers to these questions. So we want to identify and rank targets of interest. So next, here how this system works. So we run a query against PubMed, isolate publications of interest, and use generative AI to determine publications that really confirm the presence of the pattern we're interested in, and they also do the reasoning and rank publications based on certainty that they are of importance to us. And for every publication, we get the chain of causation. So we try to elicit the pathway used by pathogen here. And then we have another engine that tries to validate the result that the chain of causation was actually supported by the document itself. So we have different AI agents validating the results of each other, solving direct and the opposite tasks sequentially. And after that, we have the report. So in more detail, when we are working with PubMed, we have an overwhelming number of different inputs, and we have to very carefully design the sequence of steps we are executing to get results in the meaningful time range. So, for example, if we're addressing the task that I just formulated, we're looking for genetic mechanisms that different pathogens used to modulate the immune system response. If we start looking for all possible genes, of which there are millions, the queries will be taking forever if we try to run a query against each of 40 million of citations. So we are trying to reduce the universe of documents we have to sift through as early as possible, and then do more precise analysis in this smaller universe. So we are using a no-code cluster-based system that pre-indexes the entire PubMed articles and keeps them ready for running searches against them. And searches involve different steps here. So each of the queries is the type of search that we're executing. And the first step is to search for anything that is known through our ontologies as possible instance of immunity or immune response or host defense and so on. And we are looking for just those documents that contain the sentence of interest, and we also mark the sentence of interest. After that, we run the second query. In those articles that involve host immunity, we will be checking if any bacteria or virus or fungus and so on, any pathogen would be mentioned in the same sentence with the immune response. And we also mark those patterns. And inside those patterns, so we shrank our universe further, and we are already dealing with tens of thousands of possible pathogens. So after that, we run the most labor-intensive query where we're looking for human genes, and these are coming from ontologies of human genes, and there are lots of them. So this is why we're running it in the last step. After that, we mark all the results and get the fragments for further analysis by the AI system. So when we run the query, the system matches different abstracts from PubMed. And these abstracts are then passed to AI agents, and they start the analysis. So the first engine would be analyzing whether the discovered patterns indeed have the pattern of interest to us, whether this is not just an accidental match, because the linguistic rule was rather simplistic. We do not know whether there is the real relation between pathogen gene and the immune response. So here we are asking the system whether this is confirmed, and it sifts out those abstracts where this was just some accidental hit. Then the engine reasons and checks whether the pathogen was indeed modulating the immune response. It is providing the reasoning for that, so the analyst can verify without reading the entire abstract. We prepare a short reasoning snippet. As a third step, we ask the system to, we have a different engine that tries to identify the chain of causation. So once we rank the articles which are important and which are not, we are trying to identify how pathogen was affecting the molecular, the cellular structure, and what genes were used, what proteins, and so on. How did it get to impaired host immunity? So we are creating these chains, and then another agent is verifying the findings that we got. So on the output, we detected various pathogens, how they modulate the immune system response. what are specific mechanisms. We rank the articles, and we can also do quick filtering now, so the new researcher can come ask questions and get results quickly. So finally, the results are sent to the interactive report to facilitate the decision making. So the report looks like that, and from here we can go to the actual report and check what we obtained. So here on the top left, you see the list of different genes that we identified, and it is a long list. So again, we're looking for all possible genes that are used in connection with the action of different pathogens for modulating the immune system. So this is the list for all possible genes. And here we have various illnesses, various pathogens mentioned. The report is interactive, so in reality there are many more illnesses, and some of them are not even fitting on this screen. So what we can do, we can do searches here for specific pathogens. So you can specify, start typing, and the system will show you what you're interested in. So initially, so since we ranked articles by strongly supported, moderately well supported, and weakly supported, we can use this for filtering as well. So strongly supported relations between pathogen and gene for treating certain for modulating the immune system response are probably the well-known facts. So we can use these as a check for verifying that we do actually find this needle in a haystack. And if you're interested in treating dengue virus, you can click here and you see the corresponding fragments. You also see the sequence of actions that dengue virus affects antibody opsonized DenV colligates with LRB1, and this inhibits this expression, and this reduced ISG expression leads to enhanced dengue virus replication, and it causes the impaired host immunity. Here would be another chain of causation, which is much more complex. So it all comes from a single article. We might want to look for patterns that are not as well known. So then we would be checking all possible genes. And instead of looking at the most frequently mentioned genes, we can try and ask for something much less represented. And the system then shows you what illnesses can be, what pathogens are using this gene somewhere. And so you can click on a different one and results will be quite different. And we can go to specific articles and read them and see the reasoning why the system thought that this is important. So instead of doing that, we can do the opposite type of analysis. So for example, if we're interested in treating, say, hepatitis B virus, if we are interested in mechanisms that this pathogen is using. So you see the collection of genes for this particular pathogen, and you can look for specific chains of causation here that involve both this pathogen and this gene. So this is how the system can be used. And if you have any questions, please feel free asking them. And in the meantime, I can pass the microphone to my colleague Sam, who can show the actual multi-agent scenario that had been built in this system in no-code system PolyAnalyst. So, Sam.
Yeah, so before we do that, Sergey, there was one thing I wanted to clarify. So we started with the entire PubMed, which was 40 million records, obviously too many to run an AI on. And then we used a rules-based filter to get an initial catch of records that we could analyze with AI. Why did we use a rules-based filter instead of RAG?
RAG is dealing with embeddings, and the focus of RAG is to produce a single short answer to the user question. So here our goal is completely different. We are identifying all articles that can be potential targets for further analysis. So we need to generate a list of those targets. And when we're working with searches in the vector space, when we're working with embeddings, it is much more difficult to marry it with ontologies where we have different relations between genes. There are different namings, relations, and the same for pathogens, because as you saw in my linguistic query, I just asked to look for hyponyms of different pathogens. I was not looking specifically for Epstein-Barr virus or human papillomavirus or dengue virus or something else. So the AI, using AI on those millions of articles is just too expensive time-wise, money-wise, and it complicates the use of external ontologies and dictionaries.
Yes, I think that even embedding that many articles is expensive, not even to mention that you would have to deal with the lower retrieval accuracy of RAG.
Correct. All right.
Great. Did I share my screen successfully?
Yes.
Great. So this is PolyAnalyst. This is the framework that we use to actually construct the workflow for performing the analysis. It starts right after the initial rules-based query that produced our initial catch of articles. So here we have, let's see, 7,400 records. And you can see that this is a data frame. It's actually represented in the underlying, beneath the framework as a pandas data frame. So each record contains an abstract from the PubMed, as well as a pathogen and a gene that are potentially interacting to create an immunomodulatory response. So what we do is we take these records, we remove some duplicates, and we pass them to an AI that performs the first step of trying to judge whether or not the pathogen interacts with the gene to produce an immunomodulatory response. And this is just done with a prompt that feeds in the the title and the abstract and asks a question. This prompt is compiled on every record of the data set to produce a little distinct version of the prompt and then fed to the AI. So this is an older project. We're actually feeding it to GPT-4. And yes, so we parse the output, and we extract the level of support that is supplied for the hypothesis that the gene actually does interact with the pathogen to produce an immunomodulatory response. So in this particular node, we're just mapping the language model's output, which is a natural language output, onto an ordinal output, 0 through 3. That's a prompt engineering technique. You don't want to make the language model respond with a number. There was a study which actually found that if you ask for a response on a scale of 1 through 10, the language model produces very inaccurate answers. But if you ask on a natural language scale, say very poor to very strongly supported, the language model produces more accurate results. So we're just translating the natural language reply into an ordinal scale. That's more useful for us. We do a little more cleaning. We filter out the irrelevant articles scored as 0. No response between no relationship between gene and pathogen. And over here in the second branch, we're calculating those chains of causation. That's when the language model actually attempts to determine how the pathogen produces an immunomodulatory effect via the gene. Again, this is done using a query to the language model. We provide it with a prompt, and that prompt contains the title and the abstract, and it's asked to attempt to determine how the pathogen actually produces an immunomodulatory effect via the gene.
Sam, and can you specify what is shown in the brackets in the query? So the people are typically using prompts as pure natural language. Here you have some insertions in the brackets. So can you comment what they mean?
Yes, so this is basically a variable. It can have a different value for each column. So if we go over here, we see that there's a column called PT pathogen, and that includes the extracted pathogen for the record. When this prompt is compiled for every record in the data set, that value will just be imputed to the value to that row's value, which you can see right here.
Thank you.
That allows to create a little variation of the prompt for every row in the data set. So for instance, right here, it's going to inject the title, and right here, the abstract, and it's prompted to think about the relationship between the pathogen and the host immunity, as described in the passage, if such a relationship exists. If the passage does not describe a means by which the pathogen impairs host immunity, write no effect in place of a causal chain, or as the last link in the causal chain. So it attempts to actually produce a description of how the pathogen produces an immunomodulatory effect via the gene, which we extract right here. But sometimes the AI may make mistakes, and so we include a verification step. Basically, we just pass the proposed chain of causation to a second AI, and it's asked to check the work of the first. Does the proposed chain of causation accurately characterize the relationship between the pathogen and host immunity that is described in the following passage. Let's think step by step. This was back from before the reasoning models. Not showing the most recent work. It says you may use both recalled scientific knowledge and information from the pathogens. And again, it checks to, it says yes, no, or unsure as to whether or not the chain of causation is accurate, in order to get rid of some of those mistakes. And we find that the causal chain was found in 90% of cases and validated in 80% of cases. So then we just join that new data into the rest of the data set. This node here actually performs an SQL join operation. It's just a visual interface for performing an SQL join. So you can see this specifies the key. It's handy. And then finally, we add highlighting to the passage. It's very helpful to the human user. This is performed using again, a rules-based node. This rule just says we want to highlight the value in the column called hit, and that's the value that's highlighted. This text has special significance to the reader because it includes a description of the gene, pathogen, and mechanism that we care about. And finally, we do a little bit of visualization. So this creates a chart, a bar chart displaying the pathogens that were mentioned. And this one over here is visualizing the genes. This is then provided as an import into that web report that Sergey showed. It's a sort of it's a reporting functionality, sort of like Power BI or similar tools, all done within the PolyAnalyst interface. And that's how we created the ranked list of candidates for drug development that you see here. And now it's interesting you might be interested to note that we actually did include some positive controls in this list, so that the top candidates I think are actually known targets, just to make sure that we were able to find those needles in the haystack. It validates the efficacy of the pipeline.
All right, thank you, Sam. So I just wanted to summarize that the solution that we are demonstrating, it's an end-to-end solution that that speeds up the search for possible biological targets, and the objective is to provide systematic approach and speed up the early drug discovery process. It significantly shortens the time scientists have to spend to identify new targets. The savings might be of a couple orders of magnitude. And in addition to getting their questions answered, scientists can also see the chains of causation that might be of interest. So they see the complete genetic mechanism. They also can see things of possible importance to them highlighted in the text, which further speeds up the analysis that they need to perform. And this is just one of the applications. Here we were looking to solve a specific task of finding how different pathogens are, what mechanisms they're using to modulate the immune system response. Questions that scientists need to answer might be quite different, and having the entire solution developed easily on a single platform, PolyAnalyst, it allows them to quickly build solutions of specific interest to them. PolyAnalyst here combines different techniques for data manipulation, orchestrating multi-agent AI system. And the first step, as you remember, was based on NLP and linguistic rules. And the system had to deal with enormous number of articles, enormous number of genes, and lots of pathogens. So the initial analysis was done on a cluster of machines where the calculations are performed based on MapReduce technology. So they are doing the analysis of different portions of data simultaneously. And this enables the user to have the entire PubMed pre-processed and stored at the user location. So different queries that they need to run against this data would be taking minutes instead of hours or days that it would take otherwise if they did not have pre-indexed PubMed available on their system. With this, we probably showed you everything we have for today for this specific type of use of AI for early drug discovery. If you have any questions, please feel free to send them through chat or you can contact us by email later and we'll be happy to answer your questions. And you can download and try the system PolyAnalyst on your own from the site megaputer.com. So thanks everyone, and we look forward to talking with you again. Have a good day.